Good afternoon, everybody. Hope you guys all had a good lunch, and we are going to start the tutorial right now. So last week we talked about scientific method, which involves a process of trying to understand a phenomena that you have observed, and then you're going to conduct an experiment to test your hypothesis. And we talk about how to set up the experiment with the various controls and treatments and variables. And today we are actually going to start the more biology-focused aspect of the course, and it is called macromolecules. Okay, macromolecules. Now, before I begin, I would like to remind everybody that you should watch the lecture recordings before coming to class. Okay, so this is what I see on the on eCentennial. There are 28 of you, but only 11 of you have watched the first part, or at least clicked on it. I don't know if you actually watched the whole thing, but only 11 of you have clicked on the first recording, and then only 8 of you have clicked on the second and the third. And you know, if I go in, I could see who it is, but I'm not going to do that. My point is, you have to watch the recordings, okay, before coming to the tutorial, because the tutorial is a condensed version of what I am teaching you in the recordings. And you know, watching the recordings will give you the whole wholesome experience, the broad spectrum. Okay, the tutorial is not a substitute for the actual lecture. Okay, so I understand a lot of you you know, come from high school directly, and you know, in high school you might be used to, you know, having synchronous sessions all the time, but in this particular course, again, we have the asynchronous session, which is where you watch the recording yourself, and then we have the synchronous session, which is what we're doing right now. So next week, please watch lecture 3 recording before coming to the tutorial. In any case, we are going to do the study guide right now. Okay, so macromolecules. What are macromolecules? Well, macromolecules are really big molecules, basically, right? Macro means big. So something like water, H2O, that's a tiny molecule because it only has 3 atoms in it, right? So there are 2 hydrogens and then 1 oxygen. A macromolecule, on the other hand, instead of having 3 atoms in it, it would have like thousands and thousands of atoms. So that makes it really big. And in the lecture, we learned that there are 4 different types of macromolecules. There is the carbohydrate, there is the protein, nucleic acid, as well as lipids. Daniel asked, do these completed study guides get posted to eCentennial? Yeah, so my other class was requesting this as well. Yeah, sure, I'll post it online. Okay, but still, right, it's important to come to the tutorial as we fill it in, because you know, I tell you extra information, right? So where was I? Right, so there are 4 types of macromolecules, right? The 4 that I've mentioned. And before going into the details of what each one does, and you know, their structures and what they're made up of, let's do a little bit of basic chemistry review. Okay, so as you know, on the periodic table, there are like a little bit over 100 elements, and these are the stuff that makes up the universe as we know it. And of those elements, only 5 of them are going to be the majority of the elements, that is in living organisms, including human beings. And they are carbon. Okay, we have a lot of carbons. Oxygen, right? The chemical symbol is represented in the brackets. Hydrogen, nitrogen, and phosphorus. Okay, so these are the elements that makes up a bulk of what we are. Okay, all the proteins, all the lipids, and so on and so forth. All these are the ones that are found in high percentage. Okay, So someone asked, I thought calcium is for the last one. So calcium is actually not a major element. We do need calcium. Okay, we have them in our bones, but compare in terms of percentage, actually calcium does not take up too much of what we are made up of, and that belongs to calcium belongs to a group of elements that we actually just need a little bit of. Okay, and that is called trace elements. Okay, so trace elements means only found in small quantity, small amount in the body, Okay, so take calcium, for example, right? You might think calcium is really good. So, maybe we want to have more calcium, but actually that's not true. If you have too much calcium, your kidney could form kidney stones, and that's not a good thing. And if you have too much calcium in the blood, then you actually would have like twitching in your muscle, okay? Because to contract your muscle, you need calcium, and too much calcium is going to cause your muscle to become hyper excitable. That's what we call it, and you might have uncontrolled twitching. Right? So we don't want too much of it, and too little is not good either. Right? If you don't have enough calcium in your diet, maybe your bones will get weaker and you will get osteoporosis. So often the trace elements are a little bit tricky. Right? It's only going to be within a very narrow range. Too much or too little, that often will cause some kind of problem. So tell me, anyone else, anyone can tell me, what other trace elements are there going to be besides calcium? Anything that you read or like heard in the lecture? Yep. Okay. So very good. I have a bunch of people telling me stuff. So iodine is another one that I believe I talked about in the lecture, right? If you don't have enough iodine, you're gonna have a thyroid problem, right? Okay. The thyroid gland is where you make the metabolic hormones that controls your metabolism, and that needs iodine. Okay. And yeah, potassium. I don't think I mentioned potassium in the lecture, but you definitely need potassium. You know those sports drink that you drink after the after you exercise, right? Those have potassium in it because that's an important electrolyte, right? Okay, zinc definitely, and a bunch of other ones. Okay, so here are just like 4 examples. Moving on, and again, please feel free to interrupt me if you have any question. Okay. So there are 2 types of compound. What is a compound? When you take elements and you put them together, they form compounds. Okay. So there are 2 types of compounds that you can form: ionic compound and molecular compounds. So if you think about the periodic table, I don't have one with me, but periodic table looks something like this, with hydrogen here and then, helium over here. And then there is usually a staircase. Your chemistry teacher would tell you this, right? Ms. Chung would tell you that there is a staircase here, and on the left of it, you would have the metal, and then on the right, you would have the non-metal, right? Non-metals. So if you take a metal and you combine it with a non-metal, the result that you get is an ionic compound. Okay, so let's write that down. This is metal plus non-metal. Non-metal. Okay. On the other hand, if you take non-metals and you combine them chemically, then you will get a molecular compound. So this is non-metal, non-metals only. Okay. So types of bond formed. Bond is basically like what holds the atoms together. Okay, they're like molecular glue that glue the atoms together in a compound. So there are 2 types of bonds you can have. For ionic compound, you form what is called an ionic bond. So in ionic bond, it involves a transfer of electron or electrons, depending on what is involved, from the metal to the non-metal. Okay, so for reasons that we're not going to go into, because this is bio class, not chemistry class, you just have to believe me. Metals, they don't really like electrons. Okay, they want to get rid of their electrons, the ones that is on the outer shell. They want to get rid of it. Non-metals, on the hand, they love electrons. They're like, please give it to me. Okay, so when a metal meets a non-metal, they are perfect for each other. Okay, the metal will happily donate its electron, and the non-metal will happily accept the electrons. And that transfer of electron is what forms the ionic bond. Okay, ionic bond. On the other hand, non-metals, okay, non-metals, like I told you, they love electrons, but they're not going to transfer the electron. Okay, both non-metals would like to hang on to it. So the solution for that, instead of transferring the electron, they are going to share the electron. Okay, and they will form a covalent bond. Okay, so this is a sharing of electron. Sharing of electrons. Okay. Any questions so far? Very good. So stay at room temperature. For ionic compound, they are usually solid at room temperature. And an example of that would be something like NaCl, sodium chloride. That is table salt. Okay, that's the stuff you sprinkle on your fries. Another example would be sodium fluoride. Okay, that anybody know where do you find that? Anybody read that before? Sodium fluoride, something you have at your house for sure.
Toothpaste.
What's that? Sorry.
Toothpaste.
Toothpaste, exactly, right? So toothpaste tastes salty, a little bit salty. Not that you should swallow it, but you should still taste it, right? You could still taste it, right? So that is the sodium fluoride making that little salty taste, and the fluoride is what keeps your enamel strong. For molecular compound, it could be solid, could be liquid, or gas, right? It's possible for it to exist in one of three states. So an example of a solid molecular compound, something that's solid at room temperature, would be like C6H12O6. Anybody know what that is? C6H12O6. Yeah, go ahead. That is glucose. That's it. Okay, it's one of the monomers that we learned about. Liquid, right? H2O, that would be water. And gas would be something like CO2, carbon dioxide. Okay, so that's solid, liquid, and gas at room temperature. Okay. Now, the next thing we're going to talk about is something called polarity. Okay, what is polarity? What is polarity? So basically, polarity means are electrons being distributed evenly in the molecule? If the answer is yes, then we say it is non-polar. If it is no, then we say, like, if electrons are not being distributed evenly, then we say polar. So what does that mean? Well, H2O, as I just told you, it's a molecular compound, right? So that means it consists of non-metals only. So we have oxygen, non-metal, hydrogen, non-metals, and non-metals, they love their electrons. So they're not going to transfer it, they're just going to share it. So this bond, this covalent bond, is basically two electrons being shared. Now, depending on the atoms that are involved in the sharing process, some atoms would have a higher attraction for the electrons. Okay, we call that electronegativity. Okay, don't worry about it. If you never heard of it, you will learn in chemistry later. But basically, if one atom likes the electron more than the other one, even though they're sharing, one is kind of hogging it. Okay, it's like my daughters. When I have two daughters, I buy them a new toy. I say, please share. Okay, you have to play with it together. Okay, but then the older one kind of hogs it, even though they're sharing, but the older one is using it more than the little one. Okay, so there's uneven sharing. Okay, and that's kind of what happens with the molecule here. Okay, so oxygen is going to pull the electron towards itself. Okay, that's like my older daughter hogging the electron. So that makes it slightly negative. So this symbol is called sigma, S-I-G-M-A, and it means like slightly negative. And then it would make the hydrogen slightly positive. Overall, the molecule is still neutral. Okay, but part of it is slightly negative, part of it is slightly positive. And if that's the case, we would say water is polar, because no, the electrons are not being distributed evenly. Okay, but it's important to know, right? Water is neutral. There's no charge. Overall, there's no charge. It's just that the electrons are not being distributed evenly. Any question before I move on? Wonderful. So draw two water molecules and explain how hydrogen bonds are formed between them. Okay, so usually we draw water molecules like Mickey Mouse. And I have the circle here and then two ears. So this is the oxygen, and then this is the hydrogen. It's kind of like Mickey Mouse with no eyes. Now, the oxygen, as I told you, is going to be slightly negative, whereas the hydrogen, they will be slightly positive. So what that means is if you put it next to another water molecule, I'll draw this one upside down. And this would be slightly negative. This would be slightly positive. What happens is the slightly positive part of one molecule will be attracted to the slightly negative part of another. Okay, so hydrogen bond is basically attraction between two water molecules. Okay, where the slightly positive hydrogen of one water molecule is attracted to the slightly negative oxygen on another water molecule. Okay. And again, without going into too much of the chemistry, the hydrogen bond will occur if such interaction is between hydrogen and oxygen. And then it's also possible for other atoms to be involved, but we're not going to go into it. Okay, in our situation here, we'll just think the hydrogen bond is between H and O. So just to reiterate, do not confuse hydrogen bonds with ionic bond and covalent bond. Ionic and covalent is what's holding atoms together inside a molecule. Okay, so like, you know, in the H2O, this here, that's a covalent bond, that's a covalent bond. It is within the molecule. Whereas a hydrogen bond, it's between two different molecules. Okay, so in general, the more hydrogen bonds you have, the higher the boiling point is going to be. So for water, as you know, the boiling point is 100, okay, because it's able to form all these hydrogen bonds, right? So this H is going to be able to interact with another water molecule like that, right? Do you understand? Like this. Okay, and then eventually you're going to form like a web of interactions. Use a diagram to explain how ionic compounds such as NaCl are soluble in water. Soluble means it's able to dissolve in it, right? So if you have NaCl, NaCl, solid NaCl. So you take some salt from the kitchen and then you add water to it. What's going to happen is the NaCl will dissolve. And when it dissolves, what that means is it will dissociate. That's what it's called. Dissociate into Na plus, Aq, and Cl minus, Aq. Aq just means, Aq means aqueous, which means dissolved in water. So remember I told you, ionic compounds, the metal, in this case the sodium, likes to give away its electron. So after it loses the electron, it becomes positively charged. Right? So we call this a cation. I believe I mentioned that in the lecture. And the chlorine here is the nonmetal, which likes to receive the electron. So after receiving the electron, it becomes negatively charged. So when you put NaCl into water, what's going to happen is the Na plus will be surrounded by the hydrogen. Oh, sorry, it will be surrounded by the oxygen of the water. Because as you might remember, the oxygen is the negative part of the water. So it would love to interact with the positively charged sodium. Okay, so that's the negative part. So that stabilizes it. And on the other hand, we would have our chlorine. Chlorine is negative. So the hydrogen portion of water will be surrounding the chlorine. Okay. Now, if you have a molecule that is non-polar, something like this, like a hydrocarbon, CH3, CH2, 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 CH3, something like this. Right? This is like pentane, this was called, right? This has no charge. There's no negative, no positive anywhere. Then water will not be able to surround it. It's too big to form these kind of interactions, and it will not dissolve in water. So that's going to be hydrophobic. Right? Afraid of water, hydrophobic. Okay. So in general, if something interacts well with water, if something is able to mix well with water, we say it is hydrophilic. Okay. Almost all ionic compounds are hydrophilic. Some will dissolve better than others. And hydrophobic stuff do not mix. Okay, like you add a drop of oil in water, it's not gonna mix together. Questions before we move on? Everybody happy? Everybody with me? Good. Next, compare acid and base. Right, so what is acid and base? Well, here's a brief definition. It's just a very simple definition of acid. Karma asks, will those be on the quiz? Yes, the quiz next week. You know what? Since you asked, I might as well bring it up. If you go to the homepage of the course, you will find the quiz information. So the quiz will be Tuesday, September 21st, from 10 to 10. You can access it from 10 to 10. It will be 45 minutes. As I mentioned before, once you start the quiz, the timer will not stop. So make sure you have sufficient time alone to do it. Right? Find a place where you will not be interrupted for 45 minutes. And it will cover lecture 1, last week's stuff, and today's lecture. Okay. When it's Tuesday, you just click on this link and then you can start the quiz. In terms of how many questions, 11 multiple choice from lecture 1, 11 from today's lecture. And then for lecture 1, there will also be application questions and 3 multi-select questions as well. No application questions or multi-select for lecture 2. I'll save those for the test. 10% of your final grade. And here is a total of, what is it, 22 plus 5, 27 questions, okay, out of 30. If you need accommodation, if you have an IEP and you register with CALS, please contact me and I will give you the appropriate accommodation. Any question about the quiz? Okay. So acid and base definition. Okay, and what is the basic definition of acid? An acid is basically something that will produce H plus when dissolved in water. Safi asked, do you need to download any software for the quiz? Nope, no software needed. You just click on it and off you go. Okay. Do not do it on the phone. Okay, I have had people do it on the phone and it's just disaster. It's too small. You cannot see the whole question. Just do it on a laptop, please. Okay, or computer. Base. What is base? Base produce OH minus hydroxide when dissolved in water. And so again, these are just very simple definition of what an acid and base is. You might learn a slightly different version in your chemistry class. pH. Okay, so pH is a scale that we use to measure the acidity of a solution. So it goes from 0 to 14. If the pH is less than 7, we would call that an acid. If it's greater than 7, then it is called a base. If it equals to 7, neutral. Distilled water is neutral. Properties. Acid will be sour. Not that you should be tasting anything in the lab. And it will be corrosive. Okay. Base, on the other hand, bitter. Put a bar of soap in your mouth tonight, you will know that it's bitter. Right? Cleaning products are base and also corrosive. Examples of acid, anybody? Yep, lemon juice is a good one. Lemon juice. Stomach acid, right, or hydrochloric acid. Vinegar. Right? All these are examples of acid. Base. Base would be ammonia, which is found in a lot of cleaning product. Bleach, soap. Okay? All these are base. That's it for the chemistry. Let's move on to the macromolecules. So as I mentioned at the introduction, we have 4 types of macromolecules found in the body. We have carbohydrates, we have lipids, there is proteins, and we have nucleic acid. Nucleic acid. Okay, these are big molecules. So carbohydrates, proteins, and nucleic acids, okay, they are polymers that consist of repeating monomers. Okay, so if you think of like a train is like polymers, and then the individual parts, they are like monomers. So when you link up monomers together, you will get the polymers. And depending on what monomers you get, you use, you will get different types of polymers. Okay. So here in this diagram, you can see there are the individual monomers here. And if you connect them together, you get the polymers. Okay. So, you know, carbohydrates, proteins, and nucleic acids are not the only polymers that exist. in nature, right? Or like in the world, I guess I should say. Okay, you have a lot of polymers at your in your house. Plastic is a polymer. Polyester, right? Like your clothing is made of polyester. That's a polymer. Okay, nylon, that's a polymer. Teflon is like the non-stick stuff on your pan, right? The black coating. That's a polymer. Okay, so polymer just means it's a very long chain of molecule, and it consists of repeating units, okay, of monomers. That's what a polymer is. So in the biological system, we are looking at carbohydrates, proteins, and nucleic acids. Those are the key polymers. So how do you connect monomers together to get polymers? Well, you are going to have to do what's called dehydration. Dehydration synthesis, or also known as condensation reaction. So in dehydration synthesis or condensation reaction, you will remove a water molecule, and by doing that, you will form a bond that links the monomers together. Now, when you eat polymers, when you eat polymers, like if you have potatoes, rice, whatever, bread, then that's carbohydrates that you're taking in the form of polymers, and your body needs to break it down back into the monomer. And in order to do that, your body will have to do what's called hydrolysis. Hydrolysis. So lysis means to break. Hydro means water. So you're breaking the bond with water. Okay. So to join monomers, you have to remove a water, and to break polymers, you have to add a water. Any questions? Okay. So let's talk about carbohydrates. What elements are found in carbohydrates? Carbon, hydrogen, and oxygen. Can anybody tell me what the building blocks of carbohydrates are? What are the monomers of carbohydrates? Anybody? Yeah, okay, I say glucose, fructose, and galactose. Very good. Okay. Now I should note that these are all isomers. Okay, they are isomers. Okay, so isomer means same chemical formula. Same chemical formula. They are all C6H12O6, but different structure. Okay, think about buying a box of Lego. You build a car, and now you take it apart, and then you build a boat. The boat and the car are made up of the exact same Lego pieces, but they have different structure. Okay, so all these will have 6 carbon, 6 oxygen, and 12 hydrogen, but how you arrange them is going to be different. We call these isomers. Okay. What do we use carbohydrates for? We use it for fuel. Okay, for example, we burn glucose. Okay, but we don't like eat glucose alone, right? Like I don't think you can even buy glucose in the supermarket. I think you can buy it from specialty store. Comes in like a form of syrup or something like that. But we, no one usually would like eat glucose directly, right? We get our glucose from from food sources, and even the kitchen sugar that you have, it's not glucose. That's actually sucrose. Okay, so even though we use it as a fuel, right? We usually consume it in a much more complex form. Okay, if you drink, right? I think it's a syrup form, the glucose. If you drink a glucose syrup directly, then you will be able to get a burst of energy really quickly because you can metabolize it very quickly. But then it won't last very long. Okay. We can use it for storage of energy. So for animals, for humans, we will store it as glycogen. And then for plants, they will store it as starch. We can also use carbohydrates for structural support. And example of that would be like cellulose for the plant fibers, and also something called chitin, okay, for exoskeleton. So that's the summary for carbohydrates. Pretty straightforward. Okay, so here we have two pictures. They are both glucose. You do not have to memorize the structure of any of these. I will never show you a picture and ask you what molecules you're looking at. Okay, so you don't have to memorize them. But I will use the diagram to show you how a bond is formed. So here we have one glucose, right? If you count carbons and everything, it will work out to be C6H12O6. So I have two glucose here. And if you want to connect them, you will need to have an enzyme, right? So an enzyme is something that will speed up or catalyze a chemical reaction in the body. So if you want to connect them together, what the enzyme will do is it will remove an OH from one glucose, and then it will remove an H from the other glucose. And so afterwards, you will take away OH and H to form H2O. Okay, and then this carbon will now have one free bond, right? Because you took the OH away, and this oxygen will have a free bond because you took the H away. So this carbon will connect with the O to form a new bond. Okay, so it will look more like this. The hexagon here, I'm not going to draw everything else. Right? So this O is this O, right? And then this carbon will connect with the O on the other side. And then you will have something like this. Okay, so this here in the middle, the COC, that's this right here. And that is the new bond, okay, the new sugar bond that holds the two glucose together. Okay, and this process is again called dehydration synthesis, because you're creating something, you're synthesizing something by dehydrating it, removing water. Okay. So let's see if you are with me. Sorry, my tutorial slides are gone. So let's see if we can come back. Okay, here, let's try this one. Okay, let me launch the poll. Where's the poll button right here? Go ahead. Try this for me. Okay, only half of you have responded. I will wait a little longer, just getting a few more answers in, maybe another 15 seconds. Okay, so majority of you got it right. Okay. You have a hexagon here. Each of the corner is basically like a carbon. Okay. You will learn how to draw these next semester in chemistry when you learn organic chemistry. And then there would be like 5 carbons here. What's important is you take away the OH and the H, and then you will form a COC bond. Right? This is called an ether bond. For anybody who's interested, you don't really have to know the name. And this would be the correct answer. Okay. Good. Okay, let's try the let's keep going. Complete the reaction. So something plus something is going to give you maltose. Okay, what is that? Well, we would take a glucose plus another glucose that would give you maltose. If you look at the back of a candy bar or something, you will almost always find maltose. Glucose plus fructose, that's going to give you the sucrose. That's the kitchen sugar that I talked about. And for lactose, you would have to have glucose plus galactose. Okay. So these things on the left side here, these are called monosaccharide. Okay, single sugar. And on the right side, these things, that's going to be the disaccharide, because it consists of 2 single sugar connected together. Now, if you keep on building it, if you add more and more and more glucose, eventually you will get a polysaccharide. Okay, so one that has many sugars connected to it. So there are 4 major polysaccharides that we talked about in class: starch, glycogen, cellulose, and chitin. Okay, not chitin. Okay, chitin. That's how you pronounce it. So where can you find it? Well, starch is where you will find. Where do you find starch? Is plants. Okay, so typically in the roots. So potato, it's full of starch. It's the root of the plant. Okay, carrots full of starch. Yam full of starch, right? And a bunch of other examples. So that's where you will find the starch. The plant is supposed to store these nutrients for it to use, either to grow a new plant or to go through winters. But animals dig them up and eat them. Okay, including humans. What's the main biological function? Well, this is the storage form of energy in plants. Okay. Glycogen. Where do you find glycogen? Well, you will find them in animals. Okay, so in liver and in muscle. Okay. So if you skip breakfast this morning because you had to go to class, then what happens is you will burn your glycogen reserve to keep your body going. Okay, so glycogen is the storage form of energy in animals. Okay. Cellulose. Can somebody tell me where we can find cellulose? Very good. So a lot of people say plants. Yep. So you will definitely find them in plants, in the fibers, for example, right? Fibers. And the major function is structural support. structural support. So the plant's not gonna fall over, right? All those tree barks, all those, hard parts of the stem, right? Those structures are cellulose, okay? And it also forms cell wall. Okay, so for those of you who don't know what a cell wall is, a cell is what we are made up of, right? Plants have cells, humans have cells, but the plants, they have an extra layer outside to protect their cell. That extra layer is the cell wall, okay? And that's made up of cellulose, okay? Chitin, okay? Where do you find chitin? You will find them in exoskeleton of insect. Okay, you know how sometimes like insects, right? Like cicadas, for example. You know those like the one that goes like that in the summer. That's probably not the most accurate person impersonation of the insect sound, but the one that are buzzing around like in the summer, right? A hot summer day, right? Those things sometimes they would shed their shell, right, in order to grow bigger, right? So that thing that they shed, that's the exoskeleton. Okay, and also you will find chitin in crustacean. Okay, so crustaceans is like lobsters, crabs, right? Their shell is made up of these chitin. Okay, and it's for protection and support. Okay. So chitin is different from the other three because it's not made up of glucose, galactose, or fructose. It's made up of something called glucosamine. Glucosamine. Okay, so you might know someone who takes glucosamine, right, for their joints or for the bones. It's the same. It's the same stuff, basically. Okay. So that's it for polysaccharides. Any questions for polysaccharides? No questions. All right. Let's try this one. There are more than one correct answers, so please select them all. Okay, maybe half more minute. Right. So let's take a look at this. Select all false statement. Polysaccharides are both for energy storage and to provide structure to cells. That's correct, right? So energy storage or like glycogen and starch. Structure support would be cellulose and chitin. So that's a correct statement. And we're looking for the false one. So I'm not going to circle the first one. Glycogen is important for both energy storage and to provide structural support. That is not true. Okay, not structural support, right? Just energy storage. Cellulose is only found in plants, not animals. So that is also not correct. Glucose, fructose, and galactose are examples of monosaccharides. That is correct. So I'm not going to circle that. Starch is a storage polysaccharide made by plant cells. That is also correct. So the correct answer, as in like look for the false statement, it's going to be B and C. And those are the most popular choices. So that is good. Okay, so that's what it means by multi-select question. You're gonna have a I forgot how many, whatever I said on the quiz, like 3 or something like that. Okay, and they're worth 2 marks each. Moving on to lipids. Okay, so lipids is a little bit special compared to the other 3. Okay, so unlike the other 3 macromolecules, so carbohydrates, proteins, and nucleic acids, unlike those 3 macromolecules, lipids are not consist or are not made up of repeating monomers. Okay, instead, they consist of components. Okay. So let's take a look at what I mean by that. So there are 3 types of lipids that we talked about in class. First one is fat. Another word for fat is triglyceride, or just fat. Okay. So there are 2 types of fat, saturated and unsaturated. We'll talk more about that in a bit. But to make a fat molecule, you have 2 components to it. Okay. Component number 1 is this thing right here. That is called glycerol. Okay, and you need 1 of those. And you have these tails. Okay, that's the second component. Those are called fatty acids. And you need 3 of those. Okay. So for those of you who are interested, this is just extra information. Okay, so glycerol is it looks like this. Okay. Sometimes it's called the ho molecule. It's more like O, but you know, like Santa Claus ho, easier to remember. And then this is the fatty acids. Okay, with a bunch of carbons, could be very long, could be 16 carbon, 12 carbons, doesn't really matter. Okay, what the point is, it's really long. So if you want to make a fat molecule, you will remove the OH from the glycerol, remove the H, okay, and you take out H2O, and then you will be able to form a new bond between the carbon and the oxygen. Okay, and you would do it 3 times for each of the fatty acids, and that's how you connect them together. Okay, so it's not really repeating units. It's just like these components that are put together. Main functions. And what do we need lipids for? Well, lipids is actually useful for storage form of energy. Storage form of energy. More like long term. Okay. So like, you know, if you didn't eat breakfast this morning, I said, then you would run on glycogen, right? But if you haven't eaten lunch either, right, then maybe your body is going to start to burn off a little bit of the fat. Okay. In fact, like after 16 hours of fasting, for example, your body start to burn the fat. Okay. And if you're stranded on an island, right, then you know, as time goes on, you would be burning more and more of the fat. Okay, it's not good to burn a lot of fat in a very short period of time, because burning fat, metabolizing fat, actually makes your blood acidic. Okay, and you know, that could cause things like ketosis. You might have heard of that before. Okay, not good to have acidic blood. Fat is also useful for insulation and protection. So often you will find lipids, fats. You'll find fats around your major organs, and they will keep them warm. They will insulate them in winters. And protection, right? If you fall down, it's going to cushion them a little bit like bubble wraps. So that is lipids. Sorry, that is a fat, triglyceride. Okay, next one. What is shown in the picture here? Anybody know what that's called? No? Okay, that is phospholipid. Phospholipid. Okay. So phospholipid has three components to it. Okay. We have the glycerol like before. Just one of those. And we will have two fatty acid tail. Fatty acid times 2. And we're going to have a third portion, number 3, a phosphate group. Okay, phosphate group. So the phosphate, which is PO4 negative 3, that's going to make this part hydrophilic. Okay, so you're going to have what we call like a polar head. Polar head. And then these are hydrocarbons, and that will make up non-polar tail. Okay, so you will see later on, maybe next lecture, maybe the one after, I don't remember. You will see I draw the phospholipid like this. Okay, so here we have the hydrophilic head, and then we have the hydrophobic. Hydrophobic tails. Okay, so the molecule is very special because one part it's going to be able to bind to water, the other part it's going to be able to bind to lipid. Okay, so an example that you might be familiar with is detergent. Okay? If you wash dishes, the grease will not go away if you just use water. You have to add the detergent. So the detergent is something that looks similar to this. It will have a hydrophilic head that binds to the water, and then it will have the hydrophobic tails that binds to the grease. And then when you wash it away, the grease will get carried away with the water, because this molecule will enable the two to bind together. Right? So normally lipids and waters don't mix, but with something like detergent, something like a phospholipid, you will be able to do that. So what do we need phospholipids for in the body? Well, they actually are major component of cell membrane. That's correct. Cell membrane. Okay, we'll learn more about that, I think, lecture 4. Okay. Next, we have steroid here. Steroid. Okay, so steroid, we're gonna have these carbon rings. Okay, so that's the first thing. 4 fused carbon rings. Remember, each of the corner of the hexagon and pentagon, each of those actually are carbon atoms. So 4 fused carbon rings. And then we're gonna have these things that you stick on them. And we call them functional group. Functional groups. Okay. So think of the 4 fused carbon ring as your phone, and then the functional group would be like accessory that you put on it. Maybe you put on like a case, maybe you put on a Bluetooth or something. By adding these different functional groups to your phone, it's gonna give it like additional properties, right? Okay, so similarly, your steroid would have the core 4 fused rings, and then you would have like functional group here, you would have another functional group here, and depending on what you add to it, it's going to change its properties. So where do we find steroids in the body? Hormones, for example. Anybody know hormones? What kind of hormones are steroids in the body? I think I mentioned that in the lecture. And if you haven't watched that, you can also guess what kind of hormones are steroid hormones in the body. Christian say testosterone. That is correct. Okay, so that's the male sex hormones, but female also have a little bit of testosterone. And estrogen, that's correct. That's the main female one. Okay, so testosterone and estrogen, those are steroid hormones. We also need steroids to form water repelling surfaces. So if you think about like the waxy coating of an apple, that's the wax. It's a steroid. Like I mean, it's hard to visualize because when you hear the word steroids, you think about how people use the word steroids, right? Like you use performance enhancing drug. That's steroid, right? Okay, but you know, not all steroids are related to those kind of steroids. Okay, you know, some steroids are like the chemical definition of steroids is something that have these structures, right? So wax is an example of a steroid as well. Okay, that's it. Let's move on to saturated and unsaturated fat. So what do we have for saturated fat? What are the components? We have to have one glycerol plus 3 fatty acids. Exactly the same for unsaturated fat. The difference here is that for saturated fat, the type of fatty acids would be saturated fatty acid, which means no double bond anywhere in the structure. And it is the carbons are bound to bond to max number of hydrogen. Okay, so is there a difference between steroids and wax? Yeah, I mean, yes, there is a difference, but then for us, we don't have to really concern. The wax also has the carbon rings, but then it's the functional group that makes them, you know, different from the steroids. Hope that answers your question. Okay, types of fatty acids for unsaturated fat, we would have unsaturated fatty acids. Right. That means there might be some double bond. Okay, and it's not bonded to, the carbons are not bonded to max number of hydrogen. Okay. So it's possible for us to add more hydrogens to the unsaturated fat, and that's a process called hydrogenation. Okay, so that's how they make like margarine, for example, so that you can change it into like a semi-solid so you can spread it on your toast. That's hydrogenation. Okay. What is the state in room temperature? For saturated fat is usually solid. Okay, because saturated fat, they don't have the double bonds, so their tails are straight. Okay, and because it's straight, they can pack really close to each other. Okay, they can pack really close to each other, and that makes it solid at room temperature. The unsaturated fat, because of a double bond, right, they would have a kink in the tail, okay, sometimes more than one, and that makes them not able to come together really closely, and so as such, they are going to be liquid at room temperature. I should mention that the unsaturated fat is up to 3. Okay, they don't have to have 3 unsaturated fats on the tail. They could have just 1, 2, or all of them. Okay, so you can see here, just one unsaturated fat, the double bond creates the kink, two unsaturated fat here, okay, and then one here. Okay? But that kink really caused them to not pack together, and that's why they are liquid at room temperature. So natural sources of saturated fats, they are derived from animals. Okay, so butter, lard, right? Bacon grease, all those things are saturated fat. And you should not have too much saturated fat. Okay, if they pack easily at room temperature, they also pack quite easily in your arteries. And having too much saturated fat, it's not a good thing. Okay, so if you have to chuck down some fat, I would recommend chucking down the unsaturated fat. Okay, these are derived from plants. Okay, and usually they're liquid. So like canola oil, grapeseed oil, olive oil, they are plant derived. Now there are exceptions to these rules, right? Anybody know a plant derived fat that's actually solid at room temperature? Any chef here? People who cook? Coconut, that's right. Coconut oil smells so good, but it clots so well. It is solid at room temperature, even though it is unsaturated fat. Okay, so there are exceptions. Right? Not everything it's black and white. On the contrary, does anybody know an animal-derived fat that is liquid at room temperature? You usually feed these to kids. Butter is actually a solid at room temperature, though, right? Okay, I'm looking for an animal-derived lipid that is liquid in room temperature. Milk, good try. Milk is mostly water and proteins, though. So I know it's 3%, right? So you have some fat in it, but it's not primarily made up of fat. Fish oil, thank you. Very good. Jello, good try. Jello is actually protein and sugar. So not lipid, no lipids there, but fish oil, exactly. I don't know, did your parents feed you, or if your parent does, do you feed your kids fish oil? I remember like my mom would feed me this fish oil. Oh my God, it smells so fishy, right? Nowadays, because it's actually, I'm not joking, right? It's, no pun intended. The fish oil is actually quite fishy, but it has like good fats in it, right? It has DHA, which is supposed to be good for brain development. Okay, and they say it's good for your cardiovascular system as well. So, because of the fishy smell, instead of chucking down a spoonful of fish oil, nowadays it comes with little capsule, right? Okay, fish oil capsule, and you can buy them anywhere, Costco, Walmart. Okay, so that's that. Chocolate, yeah, chocolate again. It's there are some fat in it, and you're right. The more fat content in the chocolate, the easier it is for it to melt. Okay, but typically we would think of chocolate as solid at room temperature. Okay, very good. Let's move on to proteins, shall we? Okay, proteins. No breaks today because we have lots to cover. Sorry about that. If we take a break, we will not be able to finish. So proteins, label the different parts of the amino acids below. Okay, so proteins is a polymer, and it's made up of monomers called amino acids. Okay, so there are 20 different types of amino acids. They all have different properties. Some have similar properties, but some have very distinct properties. And they all have kind of like this similar structure. So there is the central carbon. Central carbon. And the central carbon is connected to a hydrogen always. And then there is going to be the amino group on one side. Amino group. Okay, don't worry about like why is this called amino group. It's just the way it is. Like you will learn these names in chemistry, okay? Amino group, because ammonium is NH3, so this is related to that. That's why it's called amino group. And then on the other side, over here, on the other side here, we have the carboxylic acid. Carboxylic acid. That's why this is called an amino acid. Okay, amino acid. And then there's the R. The R here is called a side chain. Side chain. Okay, and the side chain is what gives the amino acid its unique property. Like I said, there are 20 different amino acids, and you can see them in the PowerPoint slides that I have for you. So some of them are going to be non-polar, some of them are going to be polar, some of them might have a positive charge, some of them might be acidic, some of them might be basic. So there are a bunch of them. Okay. So what happens is if you have one amino acid next to another amino acid, then we can join them together with the help of an enzyme. So the enzyme will remove the OH from the carboxylic acid of one amino acid, and then it will remove the hydrogen from the amino group of the other amino acid. And from that, you will take away a water. You already know this process, right? This is called dehydration synthesis or condensation reaction. Dehydration synthesis or condensation reaction. Okay, so I'm just going to redraw this thing to show you what it looks like. Okay, so Karma asked, do we have to know all the amino group for the quiz? Of course not. What do you think? I am crazy? No, you don't have to know it. Okay, I will never ask you to name them all. So here, what will it look like? So CH and then R, we'll call it R1 for like, you know, side chain number 1. And this will be side chain number 2. And then we have the amino group here. And then this would be the carboxylic acid. You don't have to draw it, okay? Like I'm drawing it to show you a point, right? To illustrate a point. Now this carbon will now connect with this N, right? Okay, so it's gonna form a bond that will connect to this N here. Okay, so this N will now only have this H, and then you will have the central carbon, hydrogen, R2, and then C double bond O, OH. Okay, so this here, this is called a peptide bond. Okay, I want you to take a moment and compare what's on the left here and here. Do you see what I did there? Okay, so you remove the OH and the H, you take away the water, and then the C and the N like this. You see the peptide bond? Okay. Now I'm going to do this one more time for you because, like I said, I'm going to show you, I'm going to make a point at the end. So you're going to have another NH, right? And then this would be like R3, a third side chain. And then I could keep on going. I can add as many as I like, like thousands and thousands of these, hundreds sometimes, doesn't have to be thousands. Then I would have my polypeptide, right? My polysaccharide. Sorry, polypeptide, my polymer, okay, polymer, okay? So as you can see in this structure, hold on, looking for my highlighter here. You can see this here. This is just the same thing over and over again, right? Okay. That is called the peptide backbone. Peptide backbone. It's the same structure. And then the stuff here in between, right? I'll highlight it with green. So that's a peptide bond. That's a peptide bond. That's a peptide bond, right? And all these things, side chains. Side chains. Any question about what I did there? Let me move this over here. Otherwise, when I export it and put it online, it will be too, it will cause some problem. So I'll put it all the way over here, maybe. Oh boy. Maybe like that. Any question before I move on? Are we all together? Okay, I will trust that we are together. Complete the table about proteins. Elements contained in proteins. Well, we have carbon, hydrogen, oxygen, nitrogen, right? All those you can see here, plus traces of sulfur. Right? So of the 20 amino acids, there is one that's called cysteine, CYS-TINE. That one has a little bit of sulfur in it, but for the most part, it's just CHO and N. Building blocks, amino acid. And there are 20 types. Most you can make yourself, 12 of them. The other 8 you have to obtain it through diet. So we call them essential amino acids. Same thing for lipids. I forgot to mention, there are essential fatty acids, right? Omega-3, omega-6, those you cannot make yourself. You have to eat it, right? So fish, it's excellent source. Seafood, excellent source. Eggs, also excellent source of omegas 3 and 6. You can get those special eggs that are supposed to be rich in omega-3 and 6, right? But regular eggs would suffice. Biological functions. So many things proteins can do. Okay, so they can be enzymes. Right? As I mentioned, enzymes, they catalyze, they speed up chemical reactions. Okay, without enzymes, we are going to be in a lot of trouble. They can be hormones. Okay, so our body has 2 types of hormones. There are the steroid hormones that we talked about, testosterone, what's the other one? Estrogen, right? Those are steroid hormones. And then we have peptide hormones, the protein hormones. Okay, so anybody have an example of protein hormones that they know of? Oxytocin, someone said oxytocin is, I believe that is a peptide hormone, if I'm not confused, if I remember correctly. Okay, but anyone else have a peptide hormone that they know? No? No, not quite vitamin D. No. Yeah, insulin. Thank you. I think that's the example I use in class. Insulin. Very good. Okay. So insulin, we need that to absorb sugar, right? After we have a meal. Okay. And if you don't produce insulin, or if you do not have if you don't have enough adequate response to insulin, then you might have diabetes. Okay, and since someone said oxytocin, I will put that in here. You don't really have to know. You learn this next semester, okay, when you take anatomy and physiology with me. Well, I mean, doesn't have to be with me, but could be with someone else. I don't know. So oxytocin is something that helps milk production, as well as help push the baby out. Okay, so that's also a peptide. Yeah, so hopefully, next semester we will be back in campus, and it will be all weird because all of you will recognize me if you see me in the hallway, but I have no idea what you guys look like because none of you are turning on your camera, which is fine. You can keep it that way. Not gonna be, intrusive. Moving on. What else do we have use proteins for? Structure, right? Structure. Okay, so your muscle is actually made of proteins, right? Myosin and actin, those are the 2 main proteins that make up your muscle. Your fingernails, your hair, all those things are proteins. Okay, horns, we don't have horns, but like cows and goats, they have horns. Those are also protein structures. Okay, so that's that, and also fuel. Okay, so if you're stranded on an island, haven't eaten for like 4 or 5 days, okay, then you will start burning protein as well. Okay, as a secondary source of energy. Okay, now proteins are a little bit sensitive in the sense that if you expose the proteins to different temperature, different pH level, or even different ionic environment, like if you change the salt concentration a little bit, they are very sensitive to those changes. Okay, their structure might unravel, might change shape, and it will cause the proteins to not work properly. Okay, so if you remember one thing about protein and its function, it is that the shape of the protein, the conformation, that's what it's called, right? The shape of the protein, shape. Okay, the proper word is conformation. Okay, it's very important to its function. If you change the shape, you might end up changing its function. Okay, so you don't want to change the shape. And what determines the shape of a protein is embedded within 4 levels of structures. And for some proteins, it's only 3 levels. Okay, so let's talk about what that means. Primary structure. So primary structure, when you say the primary structure of a protein, it is referring to the sequence of amino acids. Okay, so like I said, there are 20 different amino acids, right? So let's say I have a protein, you know, and it has the pink amino acid followed by 2 reds, one green, blue, okay, whatever, orange, that's not even orange, but whatever. Okay, so like you have a bunch of amino acids together like that. If I read that order, right, like pink, red, red, green, green, more pink, pink, and then orange, orange, that order, that is called the amino acid sequence, the primary structure of that polypeptide. Okay, so each of these will represent a particular amino acid, right? So you might have like alanine, phenylalanine, you know, glutamic acid, whatever, so on and so forth. That specific sequence, that is called the primary sequence. Okay, so what dictates the primary sequence, right? The sequence is actually encoded in our DNA, in our genetic material. So the DNA contains the instruction, the precise instruction of what amino acids we need to add together to create the protein. Okay, so what's holding the amino acids together? Well, it's the peptide bond, right? The one that we drew earlier, right? With the backbone, that's what's holding it together. So here is the primary sequence, right? Just like a strand of polypeptide. Now, depending on the amino acid sequence, some amino acids, if you put them next to each other, they will start to coil up. Okay? Others, they will start to form these zigzag patterns. So that's the secondary structure. Okay, so they will form these alpha helix. Okay, this is alpha. And then this is beta. Right? Those are the Greek alphabet, right? Or they will form beta pleated sheets. Okay. So it will look like this. Some part will form the coil. Some part will form the plated sheets. And then some parts would just be like, you know, normal, so to speak. So whether it forms the helix or plated sheets, we call that the secondary structure. And what's holding these things together is basically hydrogen bonds formed from the backbone. Okay, the backbone is the yellow thing that I highlighted earlier, right? The backbone. Right? So hydrogen bond, basically the positive part of one portion interacting with the negative part of another portion. Okay, so that's what's forming these secondary structure. So from the primary sequence, we are going to get the secondary, and then from the secondary, it is going to form the tertiary. So this is a little bit hard to draw, but basically you will have these coils, and then it will fold back on itself. Okay, and then it will make this 3-dimensional shape. Okay, this 3D structure. And the 3D conformation, 3D shape, is what we call the tertiary structure. Okay, so that would be the overall 3D shape of the protein. And like I said, the shape is closely related to its function. So what's holding the 3D shape together? Well, this is going to be interactions between the side chains, right? The R, right? The R group. That's what the side chains are, right? So if you have a positively charged side chain on one side and a negatively charged side chain on the other side, and they're coming into close proximity, they could form an ionic bridge that will, provide support for that those two parts of the peptide, and in turns, that's going to reinforce the overall 3D shape. Quaternary structure. Okay, so that's like when I have more than one polypeptide coming together. So that's one of these, and then maybe I can take another one, and then you might have another polypeptide, right, and another polypeptide. Okay, and when they all come together, this whole thing is what we call like a protein complex. Okay, so sometimes you need to have multiple components in a protein for it to work properly, and what components you need to put together, that's called a quaternary structure. Okay, so that is the fourth level of shape. So this is when two or more polypeptides come together to form a complex. Okay, so an example of this would be hemoglobin. Right? So in the lecture I showed you, hemoglobin has like 2 alpha chains and 2 beta peptides, and they would make up the entire protein. Okay, and if you mutate just one portion of the hemoglobin peptide, that could cause you to have something called sickle cell anemia. Okay, your red cell is going to become moon shaped, and they will no longer be able to deliver oxygen for you. Okay, now it's important to know not all proteins will have quaternary structures. Insulin, for example, insulin is okay just on its own, right? It's just one peptide, and then it will stop at the tertiary level. There's no need to combine with other complexes. Okay, what's holding it together? It is the interactions between polypeptides. Okay, so earlier today, when only some of you are here, I posted a question of the week, right? So you can see that question in the chat box right now. The question says, does changing the primary structure of a protein always have an effect on the shape of the protein? Justify your answers. Now, the answer is a little bit tricky. Like you might think, yes, because if you change the primary sequence, these things are going to change, and then it's going to affect where the alpha is going to form, and so on and so forth, right? However, I also mentioned that there are many different amino acids, right? 20 of them. Some of them will share similar properties. Okay, so if the changes is that I replace one, let's say I replace one nonpolar amino acid with another nonpolar amino acid, maybe it's going to be okay. And maybe it's going to be okay because the one that we replace it is similar properties, right? So maybe it's not going to have a big effect. On the other hand, if I replace a non-polar amino acid with a polar one, and that could cause a big shift in terms of how the protein is gonna come together, right? Okay, so not always. Sometimes it's okay. Sometimes you can get away with it if the change is very similar to the original sequence. Okay, so that's the answer for the question of the week. Let's do a poll question. Go ahead. Okay. Pancreatic cells make insulin, which is a type of protein. These cells use something in order to synthesize insulin by the process of something. So what are proteins made up of? Well, I told you is amino acid, right? We just did a bunch of questions related to proteins. And to join amino acids together, we will use dehydration synthesis. So the answer is E. Okay. Monosaccharides, that's for making carbohydrates. And some of you chose B, fatty acids and glycerol. That's for making fats, right? So this is proteins. Okay? So you need amino acid and dehydration synthesis. Okay, final part here. We have the nucleic acids. So what elements are found in nucleic acids? We have C, H, O, and as well as P. All of them, all 5 major ones. Building blocks, nucleotide. Nucleotides. And the biological function of nucleic acid, they contain genetic information. Okay. And they have instructions to make proteins. Okay. Identify the phosphate group, sugar, and nitrogen base of the nucleotide. So here is the nucleotide, okay? And we have the phosphate group right here. We have the sugar group here. And we have the nitrogen base. Okay? Because you see there's nitrogen here, right? Nitrogen here. So that's a nitrogen base. So that whole thing, this whole thing is a nucleotide. Now, there are 2 types of nucleotides, 2 categories, purines and pyrimidines. So these 3 are pyrimidines. And like I said, you don't have to know how to draw them, okay? So this is pyrimidines. And these are purines. Okay. So I had trouble memorizing this when I was learning about it. And then I found out that you can memorize it like this. Pure as gold. Okay. You can memorize this phrase. Then purine is adenine and guanine. Okay. So we can see the purines, they have 2 rings. But the pyrimidine they only have 1 ring. Okay. And so what happens is, according to the base pairing rule, A binds with T, G pairs with C. So when you look at the DNA double helix, right? These are the backbones, and then these are the bases. And so you have A binding to T, C binding to G, right? So you will always have the same width because you will always have a purine binding to a pyrimidine. So across the DNA ladder, it will always be 3 rings apart, so to speak. Okay. You will not have 2 purines binding together because that would be too wide. That would be 4 rings. And you will not have 2 pyrimidines together because that's too narrow. It's just going to be 2 rings. So always a purine pairing with a pyrimidine. Okay. compare DNA and RNA structure. So DNA, the nucleotide, we are going to have the phosphate plus deoxyribose. Okay, it's a ribose sugar that's missing an oxygen and plus a nitrogen base. Whereas for RNA, we will have a phosphate plus a ribose and also a nitrogen base. What kind of nitrogen base are you going to have? For DNA, we have ATCG. For RNA, instead of thymine, we will have uracil. So AUCG. DNA are double-stranded. And so, you know, the typical thing that you see on TV, double-stranded. And then RNA, it is single-stranded. So like this. Okay. Sometimes it's possible for RNA to fold up on its own. Okay, so it will fold up into like a clover leaf shape in some cases, right? And then these would have base pairing rules like that. Okay, so something like this is called tRNA, which we will learn about later on. So that's that. Question? Okay, let's do one more poll. Okay, I see that most of you have answered already. The correct one is E. Okay, purines, they are the one that have 2 rings, double rings. So let's see what's wrong with the other ones. Adenines bonding to thymine is stronger than guanines to cytosine. I did not mention this in the tutorial just now, but I think I did in the lecture. A and T, there are 2 bonds holding it, 2 hydrogen bonds, whereas C and G, there are 3 hydrogen bonds holding it. So this is stronger than AT. And this is important for something we will learn about later on. Cytosine is purine. That's not true. Okay, purine, remember, pure as gold. So only adenine and cytosine. So that's not true. Purine bind readily to deoxyribose, but not ribose. That's not true. Okay, so A and G, you will find them to both in DNA and RNA. So that's not going to be a problem. Both adenine and thymine are purines. They're not, right? Thymines are not. So the correct answer is E. Okay, so what are we going to do next is the long answer questions. Now, obviously, we don't have time to go through all of them, so we're going to give you a moment to glance through it and let me know which one you wanna go through. Just unmute, tell me, or type it in the chat box. Excellent choice. I was going to do that if nobody say anything. Yeah, number 3 is good. And number 4 is also good. Yeah, the rest are pretty similar, right? You can just refer back to the chart that we did earlier. Okay, so let's talk about 3, and then we will talk about 4. So number 3, okay, key and lock relationship. Okay, so let's quickly talk about what an enzyme is again, and how it works and stuff like that. Enzyme. So enzyme is biological catalyst. Okay, in other words, they speed up chemical reaction. In the body. Okay. To speed up chemical reaction in the body. Now, earlier we talked about sucrose, right? Sucrose. What is sucrose? Well, sucrose is actually glucose, which I'll represent with square, connected to a fructose, which I'll represent with a triangle. Now, you look at the sucrose in your kitchen, you come back like 10 years later, if you don't touch it, you come back 10 years later, it is still going to be sucrose. It is not going to naturally break down into glucose and fructose. It's not going to happen, okay? Because if it's just sitting there, it doesn't have the necessary energy for this to take place, right? But if we eat that sugar, we put it in our mouth, within hours, it's going to get converted into glucose and fructose. How does it do that in the body? And how does it not happen in the kitchen cabinet? Well, that's because in the body, we have an enzyme that's going to help speed up this reaction that otherwise would not have happened, okay? So the enzyme that does this is called sucrase, okay? So anything that ends with ASE usually means it's an enzyme. Okay, so the sucrose will be broken down by sucrase into glucose and fructose. So in this case, the ingredients, the reactants, right? That's the enzyme is going to work on. This is also known as the substrate. Okay. So each substrate would require a specific enzyme. Okay, there's a key and lock relationship between them, meaning when you look at the enzyme sucrase, okay, it's going to have a 3-dimensional shape, right? Because it's folded up the tertiary structure, and there is going to be an active site. Okay, so this little groove here, that's called an active site. That's where the substrate is going to fit in, right? So it's going to be the square and then the triangle. It's going to fit right into the active site, okay? And what happens is, let's say this remote control, it's the substrate, and then my hand is the active site of the enzyme. When the substrate goes into the active site, it will actually cause the enzyme to shift the shape a little bit so that it grabs onto it. So this is called like induced fit, okay? So like it binds to it and then it causes it to grab onto it, make it more snug. And then after you're grabbing it, that grabbing actually weakens the bond, and then you can break the bond, and then you will release the products, which in this case is glucose and fructose. Okay, so the active site is of a particular shape, and so is the substrate. If I have fructose instead, fructose, right? Fructose would be glucose, which is represented by the square, plus galactose. This will not fit in the active site of sucrose, right? And it will not be able to help with the breakdown of fructose. And so to break down fructose, you will need to have another enzyme. And if you want to guess the name of that enzyme, it's going to be fructase. Okay. Sorry, I don't mean to say fructose. I mean to say lactose. Sorry. lactose. Okay, so lactose is glucose plus galactose. And if you want to break down lactose, you will need to have lactase, okay, the enzyme lactase. Okay? If you don't have lactase and you eat lactose, right, it might give you upset stomach. Right? You might be lactose intolerant. Right? So there's a key and lock relationship. If you change the shape of the active site, it's not going to work. It's like bending the key. It will no longer open the lock. So what are some factors, the next question, right? Number 4, what are some factors that's going to affect the shape of the protein? Factors affecting the conformation or the shape of protein. Anybody want to just brainstorm here with me? What are some factors? I kind of mentioned some during the session today. Just throw some in here and there. Anybody caught some? No? I know you guys are tired too. You guys had math this morning, right? I think. So that's like 3 hours of math and 2 hours of this. Probably is tiring. Temperature, right? If you heat it up, right, then the hydrogen bond is going to start to break down. And if you break down the hydrogen bond, then the tertiary structure and the secondary structure is going to fall apart. pH, definitely. Changing the acidity will cause hydrogen to be ripped off from the some of the side chains, or if you increase the pH, you might put some hydrogen on, and that's going to change the shape, right? So we talk about ionic concentration, salt concentration, right? Hydrophobic environment. Right? So most protein exists in an aqueous environment because our body is mostly made of water, right? So it would try to put all the hydrophilic portion on the outside and all the hydrophobic portions on the inside, hiding it. Okay, but if you put it in a hydrophobic environment, all of a sudden, then everything's going to flip inside out, and then the protein is going to break apart. Okay, so like again, I don't know if you guys cook. Some of you probably cooked. We have like 24 people here. Some of you must cook. So like if you have a piece of meat and you want to tenderize it, right? Sometimes you can tenderize it by adding baking soda, right? Does anybody know you can do that? Yes, no, maybe, I don't know. You can answer yourself, right? If you didn't know that, try it. Okay, you add some baking soda to say your pork chop or something, and you let it sit there for like not a lot, otherwise it can taste bitter, right? Like you can add just a teaspoon, mix it up, you let it sit for half an hour, and then you go cook it. Oh my God, so juicy, so tender, right? Why is that? Because the baking soda is a base, and it's gonna change the pH, and the meat is proteins, right? So that helps you break down some of the proteins, and it's going to make it tender. Paul says some people put Coke. Yeah, you can absolutely do that. And it's the opposite effect. You are now adding acid, right? Which would also help break down the proteins. Okay, but people use baking soda because number one, it's you don't have to use a lot, and number two, Coke has a lot of sugar in it, right? So it's gonna kind of change the taste of the food as well. But yeah, Coke is definitely going to achieve the same result. Kiwi, lemon, all those things are also possible. Okay, so that's that. Enough about cooking. Here, atoms can. We're gonna finish with the fill in the blanks, and I'm gonna send you off. Okay, so atoms can combine in fixed proportion to form compounds. Molecule is the smallest unit of an compound. I think the first one is molecule. I'm sorry, has been a long day. Molecules. Okay, and then the molecule is the smallest unit of a compound. Okay, so next one, atom is the smallest unit of an element. A metal and a non-metal can combine to form an ionic compound. And if it has two or more nonmetals, then it would be molecular compound. In the solution, the solute is the substance that is being dissolved, and the portion that's doing the dissolving, that's the solvent. Substances that can be dissolved in water, they are called hydrophilic. Polymers can be broken into monomers by addition of water in hydrolysis. Two glucose molecules joined in a condensation reaction to form maltose. Maltose. Hydrolysis, the breaking of sucrose, will produce glucose and sorry, I'm reading what the someone is saying. So glucose and fructose. Okay, is it number 8 solvent? Let's see, number 8. Number 8. Number 8, polymer that can be broken down into monomers. Oh, number 7, substance that can be dissolved in water are called, no, like for example, like NaCl can be dissolved in water, right? So NaCl is hydrophilic. It's something that can interact with water, right? Hydrophilic. Number 11, storage form of energy in animal. That is glycogen. Please do not confuse glycogen with glycerol. That's something that people confuse in the past. Storage form of energy in plants, starch. The fiber in plants are made up of cellulose. We cannot digest cellulose, but it's good for us because it helps with bowel movement. Main structural components of cell membrane, phospholipids. These little guys, right? With the hydrophilic head and hydrophobic tails. Testosterone and estrogens are steroid hormones formed from cholesterol. That's the starting material to make these hormones. Double bonds in their fatty acids, that would be unsaturated fats, canola oil, olive oil. Something of proteins that catalyze chemical reaction in living organism, that is enzyme. General structure of amino acid contains an amino group, and an acid, right? Carboxylic acid, and a side chain. Conformation is the shape of the protein when it unfolds because of pH, ionic concentration change, heat, all the stuff that we talked about. It is going to denature, right? Think about cooking an egg, right? The egg white, that's where the proteins are. You heat it up, it turns like opaque instead of translucent. That's denaturing it. The monomers of nucleic acids are nucleotide. For proteins, it is amino acids. By the way, like these fill in the blanks, you can change it into cue cards, right? And it's going to help with your studying. A fat molecule contains glycerol and 3 fatty acids. Alpha helix and beta plata sheets are parts of secondary structure of proteins. Last but not least, in DNA-based pairing, thymine pairs with adenine and cytosine pairs with guanine. That takes us to 227. I think that's a good good time to stop. For next week, remember there's the quiz on Tuesday. If you need extra help, please email me. I will also reply email on the weekend, okay? So you don't have to wait until Monday. And please, okay, watch lecture 3 recording before coming to next week's tutorial, okay? So have a good weekend and thanks for your attention. I will see you next week. Bye for now.